All right, so we are recording now. And I'm going to share my screen. Get this over here. You can see some faces here. All right, so you guys see my Moodle page, is that correct?
Mm-hmm.
All right, so I just want to point out this is what I've uploaded, the biological basis of behavior, and this is the PowerPoint that I'll be going over today. I did not fully review this PowerPoint right before class, and I can't quite remember if I have finished it, but I can update it if not. Right, so this is the slide I always show when we start talking about the brain, right? The brain is the most important organ in the body according to the brain, right? Our brain is what's doing that thinking. It's the one that's making our decisions. It's the one that is directing our behavior for the most part. So, of course, the brain thinks that it's all important, right? This grandiose idea. So we're going to start out talking about some techniques. How do we learn about the brain? How do we go about studying the brain? What are some different ways that we can look into the brain? And then, of course, we're going to spend some time talking about the neuron, parts of the neuron, and how the neuron works. We'll talk about the divisions of the nervous system, localization of function. So where do specific functions exist in the brain? The lateralization of functions, so that's just the fact that the right side of the brain controls the left side of the body and the left side of the brain controls the right side of the body, right? We'll talk about a little bit about the endocrine system, so that slower hormonal system that also controls behavior in addition to neurotransmitters. And then we're going to spend just a tiny bit of time looking at behavioral genetics, not too much there. All right. Yeah, these slides are not in any kind of order. I already apologize for that. So first of all, let's just talk about the brain. It is approximately 3 to 4 pounds, and we do have over 1 trillion neurons, right? And each neuron is connected to about 10,000 other neurons, right? So that's a ton of connections in our brain. And every time we learn something, we're making new connections in our brain, right? We're associating new things with one another and making those new connections. That is a sign of learning. And then we have these about 10 trillion glial cells. Does anyone know what glial cells are? What do they do?
So glial cells, I know a little bit of them. They kind of lead things throughout the brain, and then whenever they need to be there, they'll start to form into the neurons that they need to be there at the time.
Yeah, so glial cells do a whole lot. For a long time, scientists just thought that they were kind of extraneous, that they didn't really do anything. But over maybe the last decade, we've come to find out that glial cells actually have their own communication system in addition to the neuron communication system. They do a bunch of things. So, Lisa, I think you mentioned that they kind of clear out some of the waste products of the brain, right? So if we have excess
Yeah.
neurotransmitters or just some kind of like brain trash, they help clear that away.
Hmm.
They also insulate neurons. So if we talk about myelin, right, the myelinated neurons, those fatty insulated neurons, actually glial cells are what forms that myelin. So there's a lot going on with glial cells, and the more we research them, the more important we realize that they actually are. All right, so how do we learn about the brain? So originally, right, we had the Greek philosophers, and they are just asking questions, right? They, trying to reason, where does our consciousness come from? What allows us to think and to make decisions, right? All of these big overarching questions that we still kind of ask ourselves today in the field. What is phrenology? Does anyone know that term?
Um,
Does anyone see the movie My Girl? It's an old movie, very sad movie.
I've seen it.
So I don't know if you remember it very well, but Veda's in the garage and she finds this head, and it's supposed to be like the bumps in your skull are supposed to tell you about your personality, right? So you feel the bumps in your head, and that was supposed to tell you something about that person, right? So this is old, outdated. No one believes this anymore. At least I hope not. No scientist believes this anymore. But phrenology was one of the first ways that we tried to get at the brain, right? It's protected by this bone, right? It's encapsulated by our skull. And if we can't access it, what can we access? Well, it turns out we can access the skull. And so they would feel the skull, look for lumps and cracks and whatever else, and then correlate that with personality or behavioral phenomenon in that person, right? Or traits. So that's the idea of phrenology. Again, outdated. What's a lesion? What's a brain lesion?
Is that like a birthmark, kind of?
No, not quite. Thank you for that guess. So it's really like a cut, right? If we lesion something, you can cut something out, right? So this is one way that we were able to learn about brains, right? Before we had these neuroimaging, fMRI, MRI techniques, what we could do was wait until someone had some sort of brain damage, right? So they got hit in the head, or they contracted encephalitis, or some other type of disease that impacted the brain. And then we would try to correlate, okay, this is where that damage is. This is what behavioral changes we see, right? So these lesions are essentially just slicing out part of the brain. So that's one way that, early way that we were able to look at the brain. Obviously, this is unethical. We can't go around cutting out people's brains, right? That's just not okay. And we can't give them diseases, but if someone happens to present to us that has a chunk of their brain taken out. So what was the guy's name who had the tamping rod go through his skull and out the other side? Do you guys remember that case study?
I don't remember his name, but I remember what the whole thing was about.
Okay, can you tell us what it's about? What happened?
It's so a metal rod went through his skull and down to his chin, and they took it out, and he was just fine. He just
Yeah,
lost some of his personality.
except for, what was that? Say that again?
He lost some of his personality.
He lost some of his personality, right? So this happened, he was working on the railroad, and his name was Phineas Gage. So he was working on the railroad, trying to build a railroad, and he was working with explosives, and he had this tamping rod, right, to tamp down the gunpowder before they blew up this rock. And when he went to tamp it down, it ignited and shot a rod through his skull, right, like damaged one of his eyes out the other side of his skull, right, was thrown, this rod. And within like a minute or two, he sat up and started talking, right? He just had a huge metal rod go all the way through his brain, and it seemed like he was perfectly fine. So, right, so the brain's remarkable. We great important part here. His friends and family and relatives do say that his personality changed drastically with this. So he went from, here he is right here, right? So this is Phineas Gage with a picture of the rod that actually went through his skull. This is a diagram of where that rod went, right? And just remember, this flew all the way out. So here we see it depicted here, but that didn't actually happen. It didn't stick there. It just went straight through. And then here is his actual skull. So he was reported to be this very mild-mannered individual. And then after this event, reports say that his personality totally changed, right? So he became very impulsive and maybe a little bit vulgar, especially compared to where he was beforehand. So this is one of our first examples of how changes in the brain could impact personality, right? So we straight damage to his frontal lobe. So Phineas Gage here. All right. Yeah, he was level-headed and calm, foreman of the railroad crew. So this was in 1848 that this happened. Destroyed major parts of his prefrontal lobes. He became very volatile, right? So easily angered. All right. We also can talk about Broca and Wernicke, right? So both Broca and Wernicke identified areas in the brain responsible for language. So they realized that if you had damage to a very particular part of the brain, right, again, a lesion in that particular part of the brain, you might have some communication difficulties in either producing speech or understanding speech, depending on which area of the brain was damaged. So we can also look at gunshot wounds, tumors, strokes, Alzheimer's, Korsakoff's, all of these disease and wounds, right, these traumatic lesions, and get from those case study data an idea of what happens in a particular part of the brain. What is a particular part of the brain responsible for? So before we were able to internally look at the brain, this is how we did it, right? We relied on case studies of disease and lesions and birth defects. All right. Ablation just means removing part of the brain. So sometimes that happens, right? So we think this is going on, I'm just going to remove this part of your brain. Let's see what happens if you don't have this part of your brain. Can you still function? What's going on, right? And we can still do these studies with animals, mice or whatever. Obviously, again, not something ethically that we can do with humans. Unless there's some medical necessity, right? So we can still talk about severing the corpus callosum. What's the corpus callosum?
Isn't that the little part that attaches to the right hemisphere or the right lobe to the left lobe?
It is, that's right. So it is the bundle of nerve fibers that allow the right hemisphere to communicate with that left hemisphere. And in some instances of severe epilepsy, medically they will go in and sever the corpus callosum. This will prevent, like let's say you're having a seizure on the right side of your brain, it will prevent that seizure from transferring to the left side of your brain, right? So instead of having a full brain seizure, now you're only seizing on one side. So it definitely can reduce the severity, but again, it's still done today, but only in extremely severe situations, right, or cases. All right. All right, so now we can talk about some neuroimaging. Let's see what slides I've got here. Yeah. Okay, so we can talk about neuroimaging and looking at anatomy. So there are different types of neuroimaging. We can look at the structure, so that's the anatomy, or we can look at the function, so that's going to be the activity of the brain. So things like fMRI, so functional MRI, is going to look at the activity of the brain. Where in the brain, when you do a particular task, where in the brain is being activated. Whereas an MRI is just going to look at the overall structure of the brain. Are all of your structures intact, right? If we look at an Alzheimer's brain, we notice that the vacuoles in the brain, the holes in the brain that everyone has are larger, right? So, so we see that a lot with brain damage. It looks like the brain has kind of shriveled up and these holes become larger. But an MRI would tell us that, right? They would look at the anatomy or the structure of the brain. We can also talk about EEG. What's an EEG? So EEG looks at electrical waves. So this is where you put the cap on, or you put electrodes all over your skull, and we look at the electrical activity in your brain. So this type of technique, well, I've got to have slides on this. Maybe I don't, okay. So this type of neuroimaging doesn't allow us to look at a picture of the brain per se, but it does tell us about the distributed activity of the brain. So where, again, in the brain is being active at a particular time when you do something. So, and often these types of techniques are used in conjunction with one another. So maybe you do an EEG and you do an MRI, something like that. And they can tell you different information about that brain.
What's the difference between what was it, an EEG and fMRI?
fMRI. So fMRI is going to give you a picture of the brain. Here, let me just show you real quick. Right, so with an fMRI, you're going to be put in a tube, something like this, and it's going to give you sort of a heat map of where is activated in the brain, right? So it sort of looks like structure, but it's not going to give you that fine detail of structure. More, it's going to tell you, like here, right, in this particular image, this is going to be the occipital lobe. So this person is looking at something, right? So we know the occipital lobe is located there. So that's what the fMRI data look like. Let me show you. Okay, so you asked about the EEG. So the EEG is going to produce data that looks something like this, just a bunch of squiggles, right? So you put a cap on, and these are measuring electrical activity over a particular area. And there's lots of different caps. So some of them have way more receptors than others. Again, here we can do it on a baby. You can do it on, here's one that doesn't have a whole bunch of receptors. And it's just going to give you squiggle line data that tell you about the activity in the brain. But it's not going to give you precise location. So each of these is going to be summed over some spatial area. Does that help clarify?
Yeah.
And then let me show you. So again, the MRI is going to look very similar. Has anyone had an MRI or an fMRI?
Yeah.
What were your experiences with it?
It's very tight and loud in there.
Yeah, so look, you can see this picture here. There's not a ton of room. So oftentimes if you're a bigger person, it can be a very tight fit for some individuals. If you have metal implanted in your body, you cannot have an MRI. So this is a good one here because it shows if you have an MRI of your head, you will have to have this like cage over your face. And you had it. Yeah, people who are claustrophobic get in there and freak out, right? So there's a lot of drawbacks to an MRI. So this might be why, or an fMRI, either one of them. They use magnets, right? So it's magnetic resonance imaging or functional magnetic resonance imaging. So if you, I gave lots of these when I was doing research at the University of Rochester. I was responsible for taking people over and getting them hooked up and into the machine, although I did not run the MRI machine. I was in there a lot. But if you, so I had patients who had maybe some shrapnel still inside their body. You cannot have an MRI because it's a big magnet and that will pull out of your skin. Literally, it will rip out of your skin, right? There's a video of someone taking a chair into the room with the magnet, and it totally distorts the chair because the magnetic force is so strong. So there's lots of reasons people couldn't have it. I was hoping, so here we see like what the structure of the brain would look like. Notice that there's no heat map associated with these, right? So it's purely looking at the structure of the inside of your brain. Whereas that functional MRI, or functional MRI fMRI looks at the particular location. We can also talk about TMS, so transcranial magnetic stimulation. Is anyone familiar with that? I have a video I can post. So TMS is where they put magnets close to your skull. You don't have to go inside anything in particular. You're not in an MRI tube or anything, but they put magnets close to different areas of your brain, and it will temporarily lesion that part of the brain. So I often show this video of a guy trying to talk or sing while different areas of his brain are being temporarily lesioned with the TMS. So I'll post that video up underneath this PowerPoint on Moodle for you all to view. I think it's very informative. So you can kind of get an idea of what that means. So instead of actually cutting out someone's brain, we can just go in, like hover a magnet over that particular area and see what the impact is. We can also talk about CAT scans. So that's going to be an anatomy. It's similar, except it uses x-rays instead of magnets to look at the anatomy of the brain, the structure of the brain. And then one more for the activity is a PET scan. And with a PET scan, they introduce a radioactive material into your bloodstream that starts to circulate, and then we can examine that to see where in the brain we see that activity. Let's see if I can. So we're going to get something similar to that heat map, right? But it's going to be a little bit different. This one, again, the PET scan introduces radioactive material. The way I remember this is, it's silly, but it worked for me. I just think about radioactive dogs. Dogs are pets. Now I've got PET scan and radioactive material together, right? And pets are active, dogs are active. So that's my quirky, weird way that I remembered that when I was in undergrad. I talked about transcranial magnetic stimulation. I think that's the only other technology that I really want to spend time on. And like I said, I'll post that video of the investigator kind of playing around with that technology. There is a video here about CapGraw. I'm going to skip it. You're welcome to look at it. Aiden, I know we saw it in cognitive class, right? Last semester we talked about CapGraw delusion. So do you remember what it was? Does anyone remember what CapGraw is? So with CapGraw, you have a disassociation between what you see and what you feel. So this is where people think that maybe someone that they love has been replaced with an alien, right? They're like, this looks like my mom, it acts like my mom, it sounds like my mom, but in my heart, in my soul, I feel that this isn't her, right? And the video is interesting. It deals with parents and a son who has CapGraw and how they deal with that sometimes. Can be extremely challenging, right? And people who have CapGraw have committed murders. They have said, you're impersonating my mom, you're not my mother, or you're not my best friend, or you're not my wife or husband or whatever, right? And because they think that it's a fake person, they've committed these crimes, tried to injure that person just to prove that you're not really who you are. But in all actuality, it's a disconnect in our brain between the emotional centers and our visual centers. So if we look at someone you know, if you look at your mom or your dad or your sibling or your wife or your husband, you should have that instant feeling of love, right? Even if it's not really something that you're consciously aware of. But when that is severed, right, now we say, okay, well, you look like them, but I'm not having that emotion anymore. So instead of something being wrong with me, something's wrong with you. All right. The neuron. I'm sure all of y'all are probably sick of seeing neurons, but that's okay. Let's talk about them. I'm just going to ask you questions because I hope that most of you know most of this. Where direction does information flow in a neuron? From what to what?
What, axon to the dendrite, right? Or is it the dendrite to the axon?
Dendrites to axons to what? Say it again. I cannot hear you, Aiden.
That's Lauren Berg for you.
Say that again, Melissa.
Lauren Berg for you.
So we're gonna go from dendrites to axons and then down to these terminal buttons, right? Or these terminal ends. How does information travel within a neuron?
Electricity.
Electrical, that's right. And then what about between the neurons?
Chemical.
Chemical, that's right. Yep. And so what do dendrites do? What's the function of a dendrite?
To receive information.
Yep, they receive that information, absolutely. The soma, what's another name for a soma?
Cell body.
The cell body, fantastic, right? So this is where we're going to house that mitochondria, that nucleus, all of the parts that make a neuron or any animal cell function, right? That's where we've got that. We've got the axon. What's the myelin sheath do?
Well, it like insulates.
Yeah, it does, absolutely. So it insulates, and what else?
Makes the chemical reaction faster, or electrical reaction faster.
Right, so it's going to speed up that conduction. So instead of the information going from dendrite to cell body and then all the way down the axon, it's going to jump from node to node to node to node, right? So you can imagine that if you don't have to travel the entire distance, but you only have to travel this little bit of distance, is going to make that conduction much faster. So we see myelinated neurons do conduct their message a lot faster than unmyelinated neurons. And we can refer to myelin sheath as myelin, or myelin sheath, or myelination, or there's a bunch of different terms that you can use. Myelinated. And then we get to the end here, these terminal ends. This is going to be the terminal button. You can see that they're a little bulbous area at the end of the neuron. What do they do?
They release the chemicals that are the transmitters.
Yep, so they release those neurotransmitters into this gap, right? So notice that the terminal end of one neuron and the dendrites of another neuron don't actually touch one another. There's a gap between them. That gap is known as the synapse. And just real quick, we can see that before the synapse, so those terminal buttons, we can call that the presynaptic membrane, and then after the dendrites, we can refer to that as the postsynaptic membrane, right? Pre, before, post, after synapse. Right? And remember, again, we're going to have up to 10,000 connections to this next neuron. So it's not just going to be one neuron connected to another. We're going to have 10,000 neurons, right, connected to one other neuron. So this is just a very simplistic drawing of what that might look like. All right, check, check, check. What are neurotransmitters? Y'all know this. What are neurotransmitters?
Okay, the chemicals.
Yes, they are the chemicals. So the chemicals in our brain that are released by these terminal buttons into that synapse, and then hopefully taken up by the dendrites of the next neuron. Right, here are some neurotransmitters. Just a few of them. We have over 100 different neurotransmitters in our brain. And it's important to know that not all neurotransmitters impact all neurons. So in order for a neurotransmitter to work on the next neuron, right, those dendrites have to have a particular lock, right? So we can think of neurotransmitters and their receptors as lock and key. So the neurotransmitters are the keys, and then the dendrites have these little locks on them. And if you've got a square neurotransmitter and a round lock, right, a round receptor, that fit isn't going to work, and so it's not going to activate that next neuron. Also, neurotransmitters can be excitatory or inhibitory. So excitatory means we're going to pass that message on, right? So if you get some news and you're excited about it, you probably want to tell someone else about it, right? So you can think about that as excitatory neurotransmitter. Whereas inhibitory means that we're not going to pass that message along, right? We're going to inhibit some behavior. So often pain pills are going to be inhibitory type. They're going to work on inhibitory neurotransmitters. So there is a popular pain drug called gabapentin, right? So it works by blocking pain, right? It's going to inhibit that pain receptor from working. So it's not going to allow you to feel pain. All right, acetylcholine, right, is working every time you move any muscle, voluntarily or involuntarily. So this could be, you know, when you raise your hand or when you go and participate in your particular sport, anytime you move, right, acetylcholine is at work. Even with your heart, as your heart is beating, acetylcholine is helping to cause that muscle constriction, right? This is also what people inject in their face or in their armpits or wherever for Botox, right? So that causes this paralysis. So if you know anyone who's ever had Botox, right? I know people who get Botox in their eyebrows or in their face, and they're not really able to smile or move their eyebrows for some period of time after those injections, right? So they have caused acetylcholine to become inactive, right? You can't move those muscles. This is also botulism poisoning. A very small dose of botulism can cause death, and it's a very traumatic death. It causes your heart to stop working, right? So if acetylcholine is causing your heart to contract, and you have botulism poisoning and your heart can't do that, you can't breathe, and you will eventually die. So botulism poisoning, Botox, etc. Work on acetylcholine. Glutamate, right? A major excitatory neurotransmitter, extremely important for learning and memory. This is also monosodium glutamate, right? So in a lot of Chinese foods, right? They'll say, you know, maybe you'll see no MSG added, or sometimes it won't say that. That's going to be a form of glutamate, right? Very excitatory. Sometimes if people are allergic to MSG, this might cause increased heart rate or sweating, something like that. GABA, again, a major inhibitory neurotransmitter. I mentioned gabapentin. If we don't have enough GABA, so this is what I mean here, so less leads to, right? So if you don't have enough GABA, it might lead to seizures, insomnia, anxiety, or epilepsy, right? So you can imagine if you need these inhibitory neurotransmitters. If we don't have enough, then our brain is overactive. If our brain is overactive, this could lead to all of these issues, right? So seizures, this like rapid firing of our neurons, insomnia, our inability to kind of shut down our brain a little bit as we go to sleep, anxiety, again, overactiveness, epilepsy, we kind of already mentioned that one. So if you don't have enough GABA, these things can result, right? So that inhibitory neurotransmitters are extremely important. And I don't want y'all to think that they're not, because they are. Dopamine is extremely important for learning, motivation. We see addictive drugs often work on the dopamine system. Same thing with sex and video games, right? So all of those can be extremely addicting. They can provide a sense of happiness, right? Our brain is flooded with dopamine. We like that feeling. And if we get that from drugs, sex, and video games, well, then we're going to continue to do drugs, have sex, and play video games, right? Again, so you can see where this might be a motivating factor. Norepinephrine, I'm not going to spend too much time on that one. Serotonin, extremely important for sleep, arousal. Does anyone know what SSRIs are? They're a type of drug, and they really act on, they're a type of like antidepressant. So they refer, it's SSRI means selective serotonin reuptake inhibitor. What is reuptake? We haven't gotten there, but does anyone remember what reuptake is in the neuron world?
Isn't it like when it's not being used? Oh, okay.
No, go ahead.
Go ahead.
No, you got it. I'm talking too much already.
Okay. Isn't it when oh my gosh, the terminal buttons take the chemicals back from the leftover neurotransmitters from the synapse, basically?
Yeah, that's right. So it just kind of like a vacuum, it just like sucks them back up to be reused, right? So with SSRIs, they inhibit that reuptake of serotonin. This means that the serotonin can stay in the synapse longer, can work on those next dendrites longer, right? Producing a better mood. Right? So sometimes when you have depression, it is because of a decrease in serotonin. These SSRIs can help prolong the amount of time that serotonin stays at the synapse and becomes active, right? Before it's being sucked back up or reuptake from the synapse. And then opioids, painkillers, morphine, heroin, these all are types of opioids, whether they're man-made or naturally occurring, right? And they definitely work on pain. There's some addiction issues with them. They make us feel good, right? Opioids just make us feel good. That's why we see so many problems with heroin addiction or pain pill addiction. They block all of our pain, and it turns out, even if you're not consciously aware of it, your body is usually in some pain at all times, right? I'm not talking extreme pain, it might just be slight discomfort. But if we block that pain, turns out our body's like, ooh, this feels good, right? And they can be extremely addictive. You know, I mentioned, Brittany, we talked about in my class in drugs, right? My brother and his addiction issues with opioids. So it happens. All right. Everyone feel okay with parts of the neuron at least? Okay. And neurotransmitters, I've spent a bit of time going over several of these. You should be familiar with them for sure. There are, like I said, there are hundreds of neurotransmitters in our brain. This is just a select few that are extremely important, not to say that the others aren't important, right? But these are a few that we just focus on in this class. All right, so the next section really talks about how neurons work. So I'm just gonna ask you guys questions. What is resting potential? What's the definition, and then what is the number associated with it?
I know the number, it's like negative 70 million volts or something.
Millivolts, yep, negative 70
that's the word.
millivolts, that's right. That's the number. And so what is the definition of resting potential? What is it? What is negative 70 millivolts?
It's always negative, so by the time it's ready to shoot, it'll be ready to go. So like that's that resting potential.
Okay. So it's just before the neuron fires, right? So that's what we say when a neuron passes on its message. That's what we say that the neuron has fired. A neuron is sitting at -70 millivolts relative to its surroundings, right? And then as we get information, that may change. So these ion channels open up and different positively charged ions flood in and change the voltage of the neuron to allow that neuron to fire. What's threshold? What do I mean
Amount
by
of voltage it takes to fire, right?
the what? Sorry.
Amount of voltage it takes to fire.
That's right. So we're hanging out at resting potential, and we're going to get some excitatory neurotransmitters that tell us that, oh, we should pass along this message. We're also going to get some inhibitory neurotransmitters, right? Remember, we're connected to 10,000 like terminal button ends from neurons at one particular time. So we're going to get this like confusing message. Some of them say yes, fire, and some of them say no, don't fire. But cumulatively, if we sum those, we have to cross a particular point known as the threshold before that neuron is going to fire or pass along that information, right? So once we pass along, so you can imagine we're sitting here at resting potential, we get some excitatory neurotransmitters, and we start to change that voltage. Starts to go more positive from that -70, but we get some inhibitory neurotransmitters too, so it's going to bring us back down, but only when that threshold is crossed, right? So we've got more excitatory than inhibitory, are we going to send that message on to our neighbors, right, as a neuron. All right, so you cross that threshold and we can say, bam, that neuron fired, right? So it sent that message on. This act is known as the action potential in a neuron, right? So we rapidly depolarize. So we go from a -70 to about a +40 millivolts. So those ion channels open, positive ions flood into the neuron, changing the voltage. And then slowly those ion channels will close and then start pumping out the positive ions to repolarize the neuron, bringing it back to resting potential. But notice, right, so we actually dip below resting potential. So we're going to go more negative than -70. So we're going to go even more negative. So this is known as hyperpolarization. What's the point of this hyperpolarization? Any guesses?
So it doesn't shoot again.
So it doesn't shoot again, yes. What else?
So it can rest.
So it can rest, okay. Also, it helps this message being transmitted only in one direction, right? Instead of this bidirectional communication, you have to have, so if you're below -70, right, if we're at -75 or 80 millivolts, it's going to take more excitatory neurotransmitters to bring us up to that threshold, right? So we can't just keep firing our neurons over and over and over again. We're going to have a stronger stimulus to cross that threshold the next time. Yeah. So what do I mean about, what's the all or none law?
The all or none law is pretty much if the neuron passes the threshold potential, like it exceeds it, it's either going to fire with its full potential or just won't fire at all.
That's right. So it either fires or it doesn't fire. So like a gun, right? You can't half fire a gun or softly fire a gun, right? You either shoot it or you don't shoot it. Just like that. The neuron either sends its message or it doesn't send its message. There's no soft sending or half sending or something like that, right? So it's going to be similar to a light switch without the dimmer, right? So it's either on or it's off. There's no halfway in between. what is important about the rate of firing with neurons? What information does that provide us? So the rate of firing will tell us about the intensity of a stimulus, right? So if we can't partially fire or halfway fire, and we only fire or we don't fire, it's going to be the rate of that neural firing that tells us about how strong or how intense a stimulus might be. So how bright a light is versus how dull a light is. The difference between a pat on the back and a slap on the back, right, is going to be transmitted in one way by the rate of firing. Also, it can be transmitted by the number of neurons that are firing. But that rate of firing is, if we're talking about one particular neuron, the rate of firing will provide that information to us, right? So you can imagine that a neuron firing, like, obviously this time scale is wrong, but it's going to be a much weaker stimulus than one firing at a rate that's something like this, right? That's going to be a more intense stimulus. So the faster the rate of firing, generally the more intense that stimulus is. All right, and again, we have over 100 different neurotransmitters. And notice the time point of this, right? So we're talking milliseconds here from that action potential, right? We're gathering information. Only at about 2.5 milliseconds here are we starting that action potential. And by the time we get to 6.5, we've returned to resting potential. So we're, 4 milliseconds here is the rate at which this particular neuron was firing and returning back to resting potential. So very quick process. Questions? Is this sort of a review? Refresher? All right. Want to just make sure that we talked about this. Again, those myelinated neurons are going to be faster at conducting that information. Yeah, we've talked about all this. Great. Okay. We've talked about synapse. We talked about lock and key. We've talked about inhibitory and excitatory neurons. Let's see here. So this is just an example of how those neurotransmitters are transmitted down the axons, right? Going down the neuron to those terminal buttons. And then these are called vacuoles. Once they reach those terminal buttons, they kind of burst open, and these little red dots would be the neurotransmitters. Again, they're going to work as keys, and then we have our receptor sites here, those locks. And if it happens to fit, then we're gathering that information, excitatory or inhibitory information, to then decide if we're going to fire or not. Sometimes these neurotransmitters are broken down by enzymes, right? So they're kind of released into the synapse, and enzymes come around and break them down, and then those glial cells can kind of wash them away, that excess material in our brain. Other times, as we mentioned, we have that reuptake where the terminal buttons kind of vacuum up excess neurotransmitters. So after the dendrites have finished using them, they'll release them back into the synapse, and reuptake will bring them back to the terminal buttons of that presynaptic neuron to be rehoused, reused in the future. Because creating neurotransmitters is extremely time and energy intense. So if we can recycle, then that's what we're going to do. Our body is very efficient in that. So that's what reuptake is. So just a pretty good depiction of what that might look like. Make sure I covered everything there. All right, So the simplest form that, most simple form of behavior is this reflex arc. Your book does talk about the reflex arc. We've mentioned it already, right? So just having that awareness, moving your hand when you touch something and you think it's going to be hot, right? We can react before we actually have to feel the pain. And we talked about how beneficial this is from an evolutionary standpoint. We do have two different types of two different types of neurons. We have sensory neurons and motor neurons. Sensory neurons are afferent, so they arrive at the brain. Whereas motor neurons are efferent, they exit the brain. So efferent, exit, afferent, arrive relative to the brain. So two totally different types of information, right? So in order to feel something, we're using those afferent sensory neurons, getting that information to our brain. And then in order for us to react, to move, to change our position, we have to then call on those efferent neurons, the motor neurons, to help our muscles contract and move in particular ways. And then we have these interneurons that are generally located in the spinal cord, and they help speed up conduction, right? So we can have the afferent neurons sending that message to the brain, and these interneurons here help communicate with the efferent neurons, these motor neurons. So they just help speed up conduction here, and this is in the spinal cord. What else do I want to say? No, that's all I wanted to say about that. Okay. So how is the brain organized? Just make sure we're good on time. So we can talk about the color of the brain. So I'm sure everyone has heard gray matter, white matter. Do you know the difference? What's the gray matter? What's the white matter? Yeah. Any ideas? So the gray matter, right? It looks gray, right? That's what we're seeing here. This is a slice of a human brain. And the white matter here is white. So white matter, turns out, are myelinated neurons, right? So I always think of fat just being a white substance. So if you've ever cooked anything that is fatty, right, that fat is usually a white color. Myelin is an insulator, fat is an insulator, right? So white matter are those myelinated neurons, whereas the gray matter is not. So we can see differences there. We can also talk about the position of the brain. I'm not too worried about that. So we can talk about dorsal ventral or anterior posterior, medial or lateral. Not too worried about you guys knowing these particular terms. We're not going to be using them a lot in this class, but know that these are the way that we really do talk about the brain in regards to those three axes, right? Top, bottom, left, right, front, back. You good? You want me to go back? These are on Moodle, just so you guys do know that. All right. So now we get into the major divisions of the nervous system. This should be a review for all of you, I think. Natalie, have you had information on the brain before?
I have not. I've been very, not confused because you're explaining it, but no, I haven't ever really learned about the brain too much.
Okay, okay. I just wanted to check in with you because I noticed that you were quiet, and I don't know how much how many psychology classes you've had.
Yeah, no, you're doing a good job explaining it though.
Okay, and this is recorded, so I will post this on Moodle as well.
Okay.
Okay. All right. So the major divisions of the brain, we have the central nervous system and we have the peripheral nervous system. So the central nervous system consists of two things and only two things, right? It's the brain and the spinal cord. Everywhere else we have nerves, so in our fingers and our toes, whatever, right? If it's not the brain or the spinal cord, is going to be included in the peripheral nervous system. So that one's pretty easy, right? Central nervous system, brain, spinal cord, right? It's central to our body. Peripheral nervous system is everything on the outside of that. So those efferent, afferent nerves, those motor nerves, and those sensory nerves that I talked about, those are going to be part of the peripheral nervous system. We can also talk about the somatic nervous system and the autonomic nervous system. So autonomic nervous system, I think it's easier to start there, really has to do with automatic movements. Or automatic behaviors, things that are not under conscious control, right? Whereas the somatic nervous system is under conscious control. So every time if you want to raise your hand and you think, okay, I'm gonna raise my hand, right? That's under conscious control, that's gonna be the somatic nervous system. Something like your heartbeat, right? You can't readily control your heartbeat. That's going to be controlled by that autonomic nervous system, right? Automatic behavior. There's that. And the autonomic nervous system is further divided into the sympathetic and the parasympathetic nervous system. So sympathetic meaning fight or flight. What is fight or flight? What happens with fight or flight?
Does it have something to do with the adrenaline and how adrenaline rushes in, and the sympathetic part of it is when adrenaline rushes in is whether you go towards what's going on or you run away from it.
Right, so it gets your body ready for action. So the sympathetic nervous system is, when you are encountering a stressful situation, right? Your sympathetic nervous system takes over. So I think, yeah, so I'll come back to that slide and I'll go back a couple of slides in just a minute, but I just want to kind of give you guys an idea. What happens, right? So as we're preparing for action, preparing for something to happen, our pupils are going to dilate, right? Our lungs are going to relax, our heart starts beating faster, our body stops digesting food, right? So food digestion takes a lot of energy. And we need that energy to either deal with the situation, to fight it out, or to run away, right? To get out of the situation. So instead of using our energy resources to digest food, our body stops digesting food temporarily. And then we also see blood vessels constrict. So remember, this is part of that autonomic nervous system, things that are not under our conscious control. And this is the sympathetic division of the autonomic nervous system, getting us ready for that action. All right, once that has passed, so this could be even something like if you are nervous to talk in front of a group and you have to do a speech in front of class, right? You might have your sympathetic nervous system activate. Right? Fight or flight. This is a scary situation. It's a stressful situation. Your heart starts beating faster. Your pupils may dilate, etc. Right? You may not necessarily. Maybe you start breathing a little bit differently. Right? Then we have the parasympathetic nervous system. So after that threat is over, so after you've given your speech or the tiger has run away or you killed it or whatever, right? And you can return to that state of rest. So we call, I'm just going to go back real quick, sympathetic nervous system, fight or flight, the parasympathetic nervous system, rest and digest. So that's a good little trick to help remember those. Right, so that threat has gone away, so our pupils are going to constrict back to their normal size. our lungs, our breathing starts to return, our heartbeat starts to return to normal, we start to digest our food again, and then our blood vessels dilate, right? So we are using that blood can flow more freely. All right. Here is another division of this, right? So we have, it's just a visual of everything that we've talked about. Overall, we have the nervous system. This is divided into the central nervous system, the CNS, and the peripheral nervous system, the PNS. That central nervous system consists of two things and only two things, the brain and the spinal cord. Whereas the peripheral nervous system, we have two further divisions, that autonomic nervous system and the somatic nervous system, right? Autonomic, automatic, somatic is under your conscious control. And again, here we have those afferent, efferent, right? Afferent arrive at the brain. They're those sensory neurons. Efferent leave the brain. They exit the brain. Those are going to be the motor neurons. And then same thing here, right? So afferent arriving to the brain, efferent exiting the brain. And then we have the sympathetic and parasympathetic. Sympathetic, fight and flight. Parasympathetic, returning to that state of rest. So rest and digest there. So it's just a breakdown of everything that we've talked about with the divisions of the nervous system. How are you guys doing? Feel all right with this? It's a lot of information, Natalie. I know if you've never had it, it's a lot of information. I hope some of it is somewhat familiar to you.
Mm-hmm.
So let's see, just want to make sure that I've covered everything. We've got central nervous system. There's more details here than I really want to cover. This note here, I says, the brain is the consistency of soft serve yogurt or semi-soft cheese, right? It's really mushy. So oftentimes, so I've held a human brain before. Has anyone had that privilege of holding an actual brain? It's just,
Yeah.
You have? That's awesome.
Yeah.
How were you able to do that? Where did you encounter that?
So we actually in New York cut open a baby donkey because it died, and so we cut like everything open, held all of its intestines, its organs, its brain. So like it was cool, but like really gross.
Right. Yeah. It's awesome and also gross. Absolutely.
Yeah.
That's cool that you encountered one. Probably fresh, right?
Very fresh.
So a fresh brain is very different than one that you would encounter in a lab. So the human brain that I held, which, I mean, I don't know, there's just something, I don't know, there's not much that compares to that, right? So you're literally holding like someone's entire life in your hands, right? All their thoughts, all their memories. It's I don't know, it's mind-blowing to me. But what you encounter in a lab has been fixated, right? So they're harder than what you would encounter with a fresh brain. Fresh brains are gooey and soft and mushy. And if you try to hold it, it kind of like oozes a little between your fingers. So, Andrew, sorry to tell you this as you're eating.
I was eating a grape too, and I was like, I was like, it's all right.
Sorry about that. Anyway, it's super cool. If you get the chance, I highly recommend interacting with some brains. Yeah, because it's neat. All right. What else do I want to talk about before we get into this? You know, I kind of think that we've covered a lot of material, and this is probably a good place to pause, just so you guys can spend some time reviewing this material before our next class. So I'll stop here with the slides. Again, these are posted on Moodle. This lecture has been recorded. I will post. So what I do to post my lectures when I record them from Zoom, I will upload it to a YouTube channel, and I will post the link to the YouTube channel on Moodle. So if you need to review any material, it will be there, but Moodle doesn't have to host it, and so it won't be particularly slow that way. It won't take up all the bandwidth. for Thursday, I have sent out the guide for writing in psychology. The file is huge, right? So there's 7 pages initially, and then there's an example of a paper that had been written in psychology format, so APA format. And then after that, there's a ton of other material that would be helpful for you all in the future. While I do recommend you all taking a look at that material, like after the example, and the example itself will be extremely beneficial. But the first 7 pages are all I'm requiring you all to read for Thursday, and then I will post a quiz on Moodle for you all to complete about that reading. So on Thursday, we'll just quickly review the quiz. So I'll have that quiz up. It will be due before class on Thursday. And then we'll quickly review that writing guide, first 7 pages, and then we're going to continue talking about the brain. So not too much for reading, just those quick 7 pages. And yeah, getting ready for talking more about different parts of the brain.
Quick question. Is it just this packet that we have to know?
Yep.
The same packet? Okay, perfect.
Same packet. Brittany's ahead of the curve. She had to read this for another class. All right. Any other questions, thoughts, concerns, whatever else? I'm planning to meet again. I'm going to stop recording. I am planning to meet again on Zoom on Thursday. So that's where I am. All right. That's all I have.
How do you like the Zoom meetings?
It's better than getting everyone sick, but it's not my favorite. How do you like it?
It's different.
Yeah.
Not the same as in class, but
No, not at all.
I'm getting used to it now that everybody's more doing it.
Are they? Are there professors doing this?
Yeah, my
Yeah.
next class, I have one. I think I only have one in class today, and she's my only in class right now.
I don't have any in class today.
Okay. Who, Andrew, who's having in class? I'm just curious.
Annette Webster.
Yeah.
She's still doing hers in class.
Yeah. Is it a small class?
One of her classes aren't because she split the classes up, but this class is, I think, a full classroom.
Wow.
I think it's about maybe 10 or, well, however many can fit.